My name is Agape Niwuhundiro. Today I'll be presenting on the destabilizing RNL mutation human gamma decreasing, and specifically on its effect on aggregation propensity and stability, as well as how it can serve as a model for human cataract formation. Now, the lens of the human eye, which is vital for clear vision, is able to carry out its function due to a large concentration of a particular protein known as crystals. Crystals come in many forms: A, or alpha, beta, and gamma. And what these do is they establish a refractive index in the eye. Even though they're extremely durable and extremely stable, because they're only born with all crystalline we'll ever have, over time, environmental stresses such as oxidative damage can cause a change in the shape and the form of these crystallines. And what can result due to the damage is the formation of aggregates or of large insoluble masses in the eye, which can cloud the vision and cause cataracts to form. Now, what we were interested in the lab was examining the effect of a given mutation on human gamma decreasing, and specifically if we could mimic the oxidative damage that takes place over the course of decades. So we designed a mutant protein, or mutation, expressed the protein, and selected it to a series of tests. Figure 2 clearly illustrates the first part of our experiment, which involves site-directed mutagenesis. First, forward and reverse primers containing the desired mutation, so the R9L mutation, were incorporated into plasmids using many rounds of denaturation, annealing, and elongation. After this was over, when we had enough copies of plasmid with the mutation, any leftover templates that did not have a mutation were digested or were cleaved by the action of DPN1. We then took this desired mutation, this desired DNA, and inserted or incorporated it via transformation into XL10 gold cells, which are particularly good at replicating large quantities of DNA. Once they had reached sufficient density, they too were lysed, and the DNA inside was purified with miniprep and was transformed into NEB Express IQ cells, which are really good at expressing protein. Once this had reached sufficient density, these cells were lysed, the protein inside was isolated in a nickel affinity column, and then the protein was purified using dialysis. The purified human gamma decreasing was then subjected to two distinct tests. First, a thermal shift assay in order to determine its thermal stability and its mean melting temperature, and secondly, a trypsin digest, which was in order to show refolding patterns as well as if there was any aggregation patterns. Now, figures 3 and 4 present the thermal shift assay, and figure 3 shows a comparison between the mean melting temperature of our R9L mutant, of a known very unstable R14C mutant, and of the wild type human gamma decreasing. Now, in figure 3 shows a clear difference in the mean melting temperatures of the three types of human gamma decreasing, with the wild type having the highest at around 86 degrees Celsius, followed by an intermediate value for our R9L mutant, and lastly by a relatively low value for the unstable R14C mutant. And what figure 4 shows us is a comparison of the melting temperature of the R9L mutant in two different conditions, in standard conditions as well as in non-standard conditions, so in the presence of a K-tropic agent. And as would be expected, there's a lower melting temperature in the presence of a K-tropic agent. The other important set of data involved in this experiment was the trypsin digest data. And we can see from looking at figure 5 that there was much less of a difference between the wild type and the R9L samples. And it was only really at the very end of the experiment, at the 30-minute time point, that we began to see the expected difference with a lower trypsin, with a lower protein, lower amount of protein remaining for the R9L than for the wild type, whereas throughout the course of the experiment, for the entire 30 minutes, there was a consistent difference between the R14C and other two samples. The results of this experiment suggest that changing the arginine, which is charged for a non-polar leucine, had a significant impact on the stability of the protein, which is why at the higher temperatures, there was more rapid thermal denaturation, leading to a lower melting point in the R9L mutant when compared to the wild type. It's also possible that the energetic favorability of the R9L mutant is less than the wild type. Hence, refolding of the R9L mutant could be further studied in a protein concentration dependent turbidity assay, for example, to see whether in the longer run or at different concentrations there would be aggregation. Overall, the R9L mutant is promising in that while unstable, it does not aggregate immediately or in the short term, and it could therefore be used to test efficacy of drugs that target age-onset cataracts and fight aggregation of protein.